Good afternoon, everyone. Welcome back to the HBL seminar. Thank you for joining us today. Today, we are hearing from Dr. Nicolas Collao, who is currently a postdoctoral associate at the Faculty of Veterinary Medicine in the Department of Comparative Biology and Experimental Medicine here at the University of Calgary. Dr. Collao did his Bachelor in Health Sciences in Chile at the University of Concepcion. And then he continued to do a master's in human physiology in the same university in Chile, under the supervision of Dr. Juan Pablo Enriquez. From there, Nicolas went on to do a Doctor of Philosophy in the Faculty of Health Sciences in the School of Human Kinetics at the University of Ottawa, under the supervision of Dr. Michael Durisio. During his PhD, he investigated the interactions between muscle stem cells and their microenvironment following cancer treatment, as well as how exercise training may prevent or mitigate the long-term skeletal muscle deficits caused by cancer therapy. From there, he went on to do a postdoctoral research fellowship at the Faculty of Medicine in the University of Ottawa under the same supervisor. And then he is now doing the postdoctoral position at the University of Calgary Vet Medicine Department. Some interesting facts about Dr. Nicolas Collao is that outside the lab, he enjoys hiking, camping, skiing, and other outdoor activities. And then he also enjoys taking long walks and playing frisbee with his dog. Today, he will be presenting his research on the role of fibro-adipogenic progenitors in radiation-induced muscle pathology.
Well, thank you very much. It's really nice to be back here in 2018. I was here for a short period of time, like 7 months, working with Dr. Walter Herzog. So it's really nice to be back to the HBL here. So as mentioned to you, Ashna, I'm currently here working as a postdoctoral fellow in vet med under the supervision of Dr. Jeff Yurnaski, working specifically in regenerative medicine. However, today in the seminar, I'm going to talk to you a little bit about my PhD project, where mainly I'm focusing the long-term skeletal muscle effect after cancer treatment, and also how exercise training can mitigate those long-term effects. So we know that worldwide that cancer remains the leading cause of death, with approximately 18 million active cases, over 10 million deaths, and approximately 2 million new cases diagnosed just in 2021, right? However, thanks to advances in technology in the last decade, in diagnosis and also treatment, right now, 33 million people are now cancer survivors. So this is a really good news, right? And the survival rates for the most common type of cancer have been increased from 50% in 1975 to 69% in 2019. And cancer in Canada remains the leading cause of disease-related death, specifically in Canadian children, with approximately 29 to 30%, here followed with heart disease. However, the good news is that the cancer survival rate have improved such that approximately 82% of the children diagnosed with cancer will survive beyond 5 years. And one of the most, among all the type of cancer, randomized sarcoma or RMS is an intramuscular soft tissue cancer that commonly develops in young children, right? As you can see in this image of the thigh of the kid, that is a really massive tumor, right? So it's an intramuscular soft tissue cancer. Where radiation therapy and chemotherapy are type of treatment, or even a combination of both, that commonly develop or treat this type of cancer RMS. However, cancer treatment during specifically the age of period or the childbirth period can lead to long-term skeletal muscle consequences. One of them will be skeletal muscle atrophy, that is the loss of muscle mass, and also skeletal muscle fibrosis that we can define by the exacerbated extracellular accumulation in the stromal compartment of the skeletal muscle, right? And something that really caught my attention during those years that have been working in cancer stem cells and skeletal muscle is how a small tumor can have a really massive impact in the host in this case. So for example, an excessive skeletal muscle loss, fibrosis, and also systemic inflammation, right? And this is important to try to understand right now, what is the cell-cell communication in the skeletal muscle niche, specifically during and after cancer treatment, and what happened in the cancer survivors, to try to mitigate this long-term skeletal muscle effect. So it's really important to maintain proper skeletal muscle mass in cancer patients and also in cancer survivors in order to decrease this long-term effect and also reduce the possibility of secondary cancer formation. And we know that the skeletal muscle is a really plastic tissue, right? The regenerative potential of the skeletal muscle relies in a small population called muscle satellite cells or stem cells. Those satellite cells or muscle stem cells reside in between the sarcolemma and the basal lamina of the skeletal muscle fiber, right? And they're essential for development, growth, regeneration, and skeletal muscle maintenance, right? And we can observe here, in a transgenic SMI model with a green fluorescent protein that mark these muscle satellite cells that are in the muscle fiber labeled in red, and we can appreciate this multinuclear muscle fiber, right? We know that skeletal muscle fiber is a multinuclear organ. And the satellite cells in this case are identified by the expression of transcription factor PAX7. And under certain circumstances, for example, result of muscle injury, first, these satellite cells before in hemostasis are in a quiescent state or like a resting state, right? And under certain stimulus, for example, when you go to the gym or you have a muscle injury, these cells are gonna re-enter to the cell cycle, are gonna activate, proliferate, and differentiate and donate the nuclei into the pre-existing myofiber in order to perform this regenerative capacity and successful have a regeneration of the skeletal muscle. However, a small percentage, just at 20%, after the activation, they're gonna cell renew and maintain this quiescent stem cell pool for further use. And this is start with this question in our laboratory. So what are the effects of radiation in juvenile muscle, right? And we know that mechanistically, the laboratory of Joe Chakalakan in 2018 showed that specifically in a mice model of radiation, when you irradiate the muscle in juvenile mice, we observe first a reduction of muscle satellite cell and specifically their function. Also, we observe an inhibition of regeneration and a decrease in the muscle fiber size and also nuclear number. So in this case, we thought like, okay, therefore one might consider in that case that the simply transplantation of muscle satellite cells, right? In this case, will be an easy solution to this problem, right? So after radiation, we have depletion of muscle satellite cells, function numbers, right? And then we can impair regeneration. So one might think that a simple solution with just muscle satellite cell transplantation. And in this case, a nice paper of laboratory of Andrew Bragg in the United States showed that, for example, these satellite cells that are well transgenically labeled in red, right? When they are injected or transplanted into an injury muscle, we can observe that these satellite cells incorporate into the skeletal muscle, and we observe a nice regeneration after 7, 14, and 13 days, all right? However, if we isolate exactly the same cells, satellite cell labeled in red, and we transplant it in a pre-irradiated muscle following injury, we observe that the regeneration is completely blunted. Regeneration don't happen, right? And the most interesting result in this paper showed that the author found in a specific small population of satellite cells that are radio-resistant, and they express highly expression of a transcription factor PAX3, that this is a progenitors factor that is under development and fetal and embryonal development. That is really important through this transition. And this found that when they isolate specifically those satellite cells that highly express PAX3 and are radio-resistant into this injured muscle, we observe also that they contribute to regeneration, right? However, when you inject those cells, right, in the pre-irradiated muscle under injury, we observe that partially help to regenerate the muscle. So basically, these results suggest that the radiation-induced alteration in the skeletal muscle niche can contribute to this impairment of the skeletal muscle, and these alterations are not solved just for a satellite cell transplantation, right? Because there is different things that happen in the skeletal muscle niche. And beside the essential contribution of these satellite cells in the skeletal muscle in growth, maintenance, regeneration, etc., these satellite cells don't act on their own, right? And we have the satellite cell also interact with different mononuclear cell populations that reside in the skeletal muscle niche, that they're really important for all the regenerative process. For example, we have endothelial cells that are for sure essential for oxygen, nutrients, et cetera. We have cells from the immune system, like natural killers, T lymphocyte, and just pointed macrophages that are essential to get and eat all this debris during the regenerative process and clean all this, and they for sure essential to this pro-inflammatory and anti-inflammatory transition during this process. And one of the cells that we were really interested in our laboratory was this mesenchymal progenitor or fibro-adipogenic progenitors. Well, so these fibro-adipogenic progenitors are identified by their canonical marker, PTGR receptor alpha, and we can observe here. And the interesting thing about those cells is the multilineage differentiation potential. So basically, under certain circumstances, they can differentiate into myofibroblasts, expressing alpha-smooth muscle actin, right? And also they can differentiate into adipocytes in different contexts. So for example, here expressing perilipin. And we also know that these mesenchymal progenitors or FAPs from now on are essential, and they contribute to regeneration by releasing different trophic factors that activate and proliferate the satellite cells, right? However, under different pathological conditions, these FAPs or FAPs colocalized in rich fibrogenic area in the stromal compartment of the skeletal muscle contribute to this exacerbated accumulation of extracellular matrix. So basically, in homeostatic conditions, and during regenerative process, these cells help, right, to regenerate the skeletal muscle. However, in pathological condition, also they contribute to fibrosis. So in summary and gap that we know so far right now, we know that cancer survivors suffer for loss of muscle mass and skeletal muscle fibrosis, right? Radiation impair muscle regeneration through the impairment in the skeletal muscle niche, right? And the FAPs are necessary for muscle maintenance, as I mentioned before, and also contribute to skeletal muscle fibrosis. However, we don't know that question, that what is the role of FAPs in radiation-induced skeletal muscle pathology? So basically, to try to answer that question and this gap, the first thing that we did was create a relevant clinical murine model first. So basically here, we get 4 weeks old mice, we apply radiation, the rest in just one hialine, the rest of the body was left chilled, so we avoid bone marrow transplant. And then we sack the mice at different time points to see acute and long-term effect, right, of the skeletal muscle. So in the morphology first, we observe a decrease in the cross-sectional area at 3, 14, and 56 days post-radiation exposure. And 56 days, or this long-term effect, is when the mice already is full mature, right? So basically, we are translating this, what happened in a children with a randomized sarcoma when we get treatment, and after that, we observe the long-term effects. And also, we observe an increase through trichome staining intensity, increase in skeletal muscle fibrosis at long-term effect. So basically, this model can replicate exactly what is happening in juvenile kids that are undergo to cancer treatment. So another thing that we were really interested in is what are the transcriptomic changes that could occur following radiation exposure. And for that, we apply what is called single-cell RNA sequencing. This is part of the 10x genomic technology that basically, and in your simple words, we can capture hundreds of thousands of cells in an oil droplet, right? And what is called an Magellan emulsion bits, right? This gel emulsion bit can contain a specific 10x barcode specifically for one specific cell, a unique molecular identifier, and a poly detail that will be essential for hybridization during the amplification period, right? So basically, after our cell homogenate, when we can digest our muscle, we pass those cells in the 10x genomics, we can have a single droplet with one single cell, right? And after that, we do hybridization, reverse transcription, and amplification. We have a period of library preparation, and after that, we sequence all our cells in order to get our data. And I know that maybe not too many people are familiar with this technique, so for a demonstration, so you can have actually understand the potential of the cell, I wanna do a small demonstration. So basically, you can screen the first name, the first letter of your name, right? So basically, my name is Nicolas, and I'm gonna screen the letter N. Are you ready? So 1, 2, 3. All right, a little bit louder. All right, 1, 2, 3. Okay, perfect. So staying here, it's really difficult to understand all the names. So basically, I could catch maybe an L, maybe another N. However, it's really important to understand in this context what happened, for example, with the J's, with the X, or with the other letters, right? So basically, for me, it was really difficult to understand this. And you have to, right now, see that you are the skeletal muscle image. So during different regenerative process or during injury and different pathology, exists an active communication within different cell types, right? So what this technology does in this case is gonna go and each of you and try to decipher what you're telling to me, right? And after that, through different computational methods and algorithm, I can extract all the information of individual cells and try to replicate this at a single cell resolution, right? So basically, we can observe this in this what is called UMAP. So basically, each cell has 1,000 of transcript, right? And we can process this just in an Excel sheet, right? So basically, we have to try to observe all this and plotted this in a 3D model. So basically, we have this UMAP when based on the different transcript of each of you or the different cell type in the skeletal muscle, we can observe that each cell have a different transcriptional signature, right? So basically, in our single cell RNA sequencing analysis, we observed that we have for sure different cells in the skeletal muscle. For example, we have endothelial cells, those FAPs, muscle satellite cell, pericyte, different cells from the immune system, et cetera, et cetera. And we observed in this heat map that each cell have a different transcriptional signature. So perfect. We get those cells and we can study what are the transcriptional processes. So basically that right now we have this model, right, that we can recapitulate on those morphological changes that happen in follow radiation exposure, for example, in a kid that have a randomized sarcoma, that we observe that we have first a decrease in the cross-sectional area and also an increase in skeletal muscle fibrosis. We want to see specifically what is the effect of your radiation exposure in the FAPs, in the FAPs, right? So our first question is, okay, what happened with the proliferation state of the FAP following radiation exposure? So basically to address this question, we irradiate in vivo, we extract our cells, in this case, through a technique called fluorescent activated cell sorting, right? And we were able to, in addition to capture all the cells, and the proliferation state with did it through an EDU assay that basically EDU is a thymine analog that actively incorporate in the highly divided cells. So, and you can see this moving. And we observed that the radiation, even at 56 days, partially decrease the proliferation of state of these FAPs or this mesenchymal protein, right? And one of the features of the radiation exposure is cell damage. So to understand that also, we observe an increase in the cell damage by a marker of a protein called gamma H2AX. And we observe the accumulation of this protein in the nuclei, specifically in different foci of the nuclei of those mesenchymal progenitors. And we observe an increase in DNA damage at 15 minutes and even 24 hours after follow radiation exposure. And another characteristic story of, or one of the answers of DNA damage could be cellular senescence, right? And the cellular senescence we can define it as cells that cannot divide anymore, but they're not dead, right? And in the past, different scientists thought that when a cell enters to a senescent state, they're just garbage cells, basically, that don't have any physiological contribution or they're not active cells. However, we know right now that those cells that when enter to a senescent state actively can release different trophic factor to the medium, right? Specifically pro-inflammatory and pro-fibrotic factor, or what is called SAPs, right? And we were able to identify that after radiation exposure, we observe an increase in the senescent state of our FAPs, but staying in beta-galactosidase. However, we were a little bit further, and the thing that we did was a senescence score. So basically, we take a few hundred of senescence gene markers, and we map it back to our single-cell RNA sequencing. And we noticed that also these FAPs and the FAPs are the highest one that express the senescent-like molecular phenotype following radiation exposure. Next, we thought like, okay, what happened with the cell count? What happened with the number? So we did an experiment by flow cytometry analysis. And in this case, we have our lean negative. We have here our muscle satellite cell expressed by this integrin alpha 7. And also we have our mesenchymal progenitor by their marker PTGF receptor alpha. And we were able to notice that, just I'm telling the story of the FAPs, that following radiation exposure, we observe a decrease in the numbers of these mesenchymal progenitors. We also validate this by immunofluorescence, and we observe here that this images, we have the skeletal muscle fiber stained with PTGF receptor alpha and DAPI marking the nuclei, and also we observe a decrease at 14 days and at 56 days following radiation exposure, similar that we observe by flow cytometry analysis. And interestingly, also we have some human muscle, in this case, working with the Ottawa Hospital. And in this case, these are not just pediatric samples. There are a variety of different human samples here, but different markers, for example, CD90 and PTGF receptor alpha, we were able to identify a similar trend to decrease. So basically, those FAPs in the skeletal muscle in humans also decrease follow cancer treatment by 45%. So basically, still we're collecting more human samples to increase the N. And right now we want to observe what are the transcriptomic changes occurring following radiation exposure in those FAPs. So basically, by a subclustering our mesenchymal progenitor for this big UMAP, we observed that the FAPs, we were able to identify 6 different subclusters. I'm not going to name all of them for sure. However, we observed that it's a change in the transcriptomic signature of all of them. Interestingly, that at 56 days post-irradiation, we observed first that the CXCL14 FAPs that are more stem-like cells in the skeletal muscle during homeostasis decreased from 32% to 19%. However, these two, specifically the DLQ1 and TNM2, that also were identified in different pathological conditions, were increased following radiation exposure. And more interestingly, also, we did a flow cytometry analysis based of regenerative FAPs that express in this case CD55, and also more fibrotic FAPs expressing CD142, their markers. And we observed that at 56 days post-irradiation, we observed a completely shift, an increase in the percentage to 21.5% to 46% in these pro-fibrotic FAPs following radiation exposure. So basically, this data is suggesting right now that following radiation exposures, these FAP are acquired this pro-fibrotic or pro-fibrotic phenotype, right, or are transitioning or differentiated into these myofibroblasts that could contribute to this long-term skeletal muscle effect that we observe in human patients. And similar that we did with the senescence scoring, we took 700 genes, in this case, related to fibrotic tissues, and we map it again back to our single-cell RNA sequencing, and we observe that FAPs are the most fibrotic phenotypes, right? And Understanding a little bit more this process, also we did a genetology analysis based on the different biological processes that occur after, or in this case, at early time points, following radiation exposure. And we observed that actually the FAPs, the biological term increases in fibroblast proliferation, different kinase activity, and also stress-induced premature senescence state. Similar that we observe with this beta-galactosidase staining. And another, when we have for sure transcriptomic changes, we can have different morphological changes that can occur in different cells. And in this case, also we were able to identify that after radiation exposure, we observed an increase in the area of these FAPs, and also more structure or more stress fiber formation of alpha smooth muscle actin after radiation exposure, and we have our positive control for sure with TGF-beta1, suggesting that they acquire this fibro-adipogenic phenotype. And for sure, we did a Western blot also that these phospho-MAT signaling were increased after radiation exposure in those cells. And the thing that we did through our bioinformatic analysis was what is called a cell chat. So basically, we're really interested to know what is happening within what cells are talking, right, during this context of irradiation. So interestingly, we observed first that TGF-beta, this transforming growth factor beta signaling pathway, was the highest one in the different that change following radiation exposure at 56 days post-radiation. We validate this by immunofluorescence analysis, and we observe an increase at TGF-beta1 based on the immunofluorescent intensity in the stromal compartment of the skeletal muscle following radiation exposure. And right now we want to know the ligand receptor pair interaction, right? So basically we took all the mRNAs, we do our bioinformatic analysis, and we observed also that TGF-beta1 was the main signaling pathway that go through different receptors, TGF-beta1 and TGF-beta receptor 2. And interestingly, when we observed this cell chat analysis, we found that monocytes and macrophages were the main senders of TGF-beta1, this profibrotic signaling, and actual FAPs were the main receiver in this case, right? And for sure, we observed that this goes through TGF-beta receptor 2, and we can validate it through, in this case, through a Western blot. So what we know so far in this case, radiation depletes FAP with a relative sparing of fibrotic subpopulation. Radiation alters FAP's proliferation and induce senescence state, right? And radiation induces a profibrotic differentiation of FAP through TGF-beta1 signaling that was mainly the outgoing signaling by monocyte and macrophages. And right now we want to know the functional consequences following radiation exposure in this specific mesenchymal progenitors, right? So basically here, we isolate FAPs and muscle satellite cells through this technique that I mentioned to you before that is called fluorescent activated cell sorting. We played our FAPs that were in our control or irradiated, right? We let them grow, and then we took the conditioned media of those and played it during differentiation of these muscle satellites, right? To observe the paracrine effect of the irradiated FAPs through these muscle satellite cells. And we observed that when we applied the conditioned media of the irradiated FAPs, we observed a decrease in differentiation index of myotubes and also a decrease in the fusion index of this myotube. And we observe here this by staining of myosin heavy chain. So basically right now we understand that these mesenchymal progenitors or FAP can contribute to the skeletal muscle fibrosis, but also through the release of different trophic factor in these pathological conditions can decrease the cross-sectional area or induces skeletal muscle atrophy. And we know that exercise is really good for cancer patients and also cancer survivors, right? And however, the combination of resistance and endurance exercise training is a non-pharmacological modality that can for sure induce an increase in cardiovascular fitness, increase muscle hypertrophy and strength, and also increase skeletal muscle adaptation that ultimately will lead to a better quality of life of cancer survivors, right? Unfortunately, this type of exercise training, the combination of resistance and endurance exercise training, all the research have been mainly done in adult cancer survivors, leaving aside what is happening in juvenile cancer survivors. So we try to address this gap also. And for that, we apply a specifically resistance and endurance exercise training modality in mice. So basically here we get 4 weeks old mice. We inject them with this randomized sarcoma, so this intramuscular soft tissue cancer, right? We apply a combination of chemoradiation therapy. So basically we did a vincristine injection. And after that, we applied 5 times 4.8 rays of localized radiation, right? And after this, we divided in 2 different groups. We have a sedentary group, and after that, we apply our resistance endurance exercise training modality. So basically here, the mice, we put a wheel in the cage of the mice, and we apply, in this case, a specific magnet into the wheel, right? So right now we have this unbalanced wheel that the mice, each time that want to run, have to counterbalance the weight of the magnet. And in this case, since it is a progressive training, up to 8 weeks, we increase the grams of the magnet. So basically, we have this endurance exercise and also this resistance exercise. Most importantly, what we observe here is that the mice that go under this training increase the total distance runnings compared to the sedentary group and also reduce the body fat formation. Most interestingly here, we observe that exists a trend to the RMS plus treatment decrease the muscle weight, right? And specifically here, the gastrocnemius. However, the resistant endurance training trend to increase the muscle mass to rescue this loss of muscle mass through treatment. And we observe also similar thing that in this case, that resistant endurance exercise training increase the total fiber cross-sectional area. Right now, going specifically, a little bit more specifically, what is happening in muscle fiber typing, we observe also an increase in type 2A fibers and also an increase in type 2B fibers follow resistant endurance exercise training. All right, now we want to understand a little bit more what happened with this mesenchymal progenitors, right? So basically, we perform a flow cytometry analysis, and we observe that actually the RMS plus treatment in this case, when you go under this specific exercise training, kind of rescue the number of mesenchymal progenitors that were lost during the treatment. And after that, we took what is called a nanostring analysis that basically we took a fibrotic and inflammatory panel and we match it and we map it through, in this case, to our cells. And we observe a difference between these RMS that undergo to the training versus the sedentary. And we observe here that specifically a downregulation of different profibrotic and cancer signaling pathway that go decrease the direct enrichment score when they go to resistant endurance exercise training. So basically, we suggest that follow exercise training, we can mitigate the inflammatory process and fibrotic process, and also potentially we can mitigate the differentiation of these mesenchymal progenitors in active myofibroblasts that can release specifically a lot of extracellular matrix. So in summary, here, part of my PhD project, we observe in this case that radiation therapy can alter the whole skeletal muscle nature, not only the FAPs, but also these muscle satellite cells, endothelial cell, and also for sure we have data of different immune cell compartments. We observe that also radiation therapy increased this fibrotic lineage or fibrotic lineage commitment of these mesenchymal progenitors, that they release a lot of extracellular matrix. And also we observe in this case, when we apply a resistant endurance exercise training, potentially we can mitigate the skeletal muscle atrophy and we can reduce this fibrosis in a context of cancer treatment. So I want to thank my ex-laboratory of Dr. Michael Delicio at the University of Ottawa, my old lab mates, all the different laboratories that contribute to this project and the different papers, the funding agency, and also the funding agency that I'm currently right now. And also want to show here that I'm really thankful to be back here in the Human Performance Lab, and also as in the last talk of team retirement, he say it's not just about papers or science, it's just about people. And I have 7 months here that really mark my transition in how to do science, because I think so the HPL have a great people here. And thanks God, all those people already here, international students, are currently friends of mine that when, for example, I have to travel to different conferences in Europe or whatever, I visit them. So it's really nice to see this, specifically this camaraderie in this context in the HPL. So thank you very much, and happy to answer any questions.
For questions, would like to start us off?
Great presentation, really awesome visuals. One thing I saw or sort of know more about, I guess, compared to the sequencing and everything, but your UMAP plots. I'm kind of curious, maybe I missed it, but what was the use of those UMAP plots? For your project? Were you able to like create the clusters based on the sequences based on that, or what did it tell you?
Exactly. So basically, the UMAP is the kind of the base analysis that we can do when we sequencing our specifically our samples. So basically, what does it mean is since we have thousands of millions of transcripts, we have to try to visualize them, right? And what this means, we collapse all the transcripts together, and the transcripts that are similar, and since we have in the gel emulsion be the specific barcode for a specific cell, we can map it back at a single cell resolution, and each cells that are clustered together means that are exactly the same cells, right? And after that, we can say, okay, what are specific signatures of this specific cell? And we know, for example, an endothelial cell highly express CDH5 or PKAN1, that is CD31. And we know that when plug this gene, it's going to be just an endothelial cell, for example. When we map again PDGR receptor alpha or SCA1, we know that highly expressed in those mesenchymal progenitors, or for example, PAX7 just in muscle satellite cells. And after that, we can understand first what happened in the percentage changes, what happened in the number, or where transcriptional changes that occur. Something that I didn't show here, we can also observe different changes, not only at the gene expression, but also we can pseudo know what is happening at the level of transcription factor, but different packages in up, right? So it's really interesting to try to understand this. And also I mentioned, as I showed it to you, we can see how the cells communicate in different contexts, right? In pathology, during hemostasis, or for example, after an injury, whatever, we can observe what is happening at that level.
Yes. Nice talk, Nicolas. Thank you. So these cells today, so they are mesenchymal cells. So they develop into another type of cells like fibroblasts and then adipocytes.
Yeah. So those cells are from mesenchymal origin, right? And really similar at the transcriptional level to fibroblasts, right? However, under certain circumstances or conditions, they can differentiate. That's why there are multi-lineage differentiating potential that they have, right? So for example, they're called fibro-adipogenic progenitors because in 2010, a group of UBC for fibrosis lab and also a Japanese group, they identified and they noticed that a specific cell can differentiate specifically in these two lineages. But right now we know that also those cells in different conditions, in vivo and in vitro, can differentiate into osteoclasts and other cell types, right? Depending on the condition.
So your study is mainly done before they differentiate, or you know that they have differentiated into fibroblasts, and now they're producing lots of collagen.
Yeah, so we want to understand what what is the main driver of this skeletal muscle fibrosis prior, because we don't know what was happening. So basically, all this research, we noticed that actually those cells are the key cellular player that contribute to this skeletal muscle fibrosis by changing the morphology, increase the extracellular matrix deposition, they go to a senescent state that releases different pro-inflammatory and fibrotic, also cytokines, chemokines, and also they contribute to decreasing skeletal muscle mass through the inhibition or reduction of fusion index and differentiation index of these myotubes.
Okay. And if I may, so you talked a lot about fibrosis,
Yeah.
but what about fat infiltration and the accumulation of fat
Yeah.
in the muscle after the
Specifically, at this type of pathology radiation, we didn't observe, we did also pure lipid staining, if they go through adipogenic differentiation, but we didn't notice that. We also, no significant changes in pure lipid staining, they don't differentiate that much into adipocyte. However, they differentiate, at least in this pathology, in this context, into myofibroblasts. However, in different pathological conditions, for example, there is a different Duchenne muscle dystrophy or a different dystrophies, they can differentiate into adipocytes too and contribute to this fatty infiltration that we observe in the pathology at large.
Do you know why?
In the fibroblasts,
Yeah,
or
why they develop into one type of cell and not the other?
it is different context. I think so. The most important thing here is what they sense in the skeletal muscle image, right? In different pathologies, for example, Duchenne muscle dystrophy, right? We have a constant rupture of the sarcolemma and the muscle membrane all the time, right? And we have also an excitation of a muscle satellite cell. However, radiation is it's not a normal type of injury because they go through all the cells without damages too much the membrane of the muscle of the myofiber. However, they damage specifically the DNA of the cells there. For sure, I think so. What is marking what cell is going to go through one lineage to another is what is happening in the microenvironment at the pathological conditions.
So one follow-up as well is, so after radiation, there is no accumulation of fat.
We didn't answer that.
Earlier?
Oh, yeah, great, good talk, congrats. And related to Ina's question, did you, or maybe you explained it, I missed it, but did you look for the transcription factor that drives that differentiation of the fibrotic phenotype? Like, is it known, or did you look at it in your transcriptomics?
Yeah, we didn't do that because one of the limitations that we get a lower number of cells in that specific context. However, during another project, when we induced cancer cachexia, in this case, injected LC and induced specifically cancer cachexia in loose mass at the early time, but we try to decipher specifically what transcription factor is driving these changes in thinking.
Oh,
Thank you, Nicolas, for your talk. I am wondering about I have two questions. What is the magnitude of the damage or fibrosis damage that the radiation produce in terms of percentage? Because sometimes when we are analyzing this, histopathology images, we have to set some percentage of damage or changes. So what is the value that you have in your?
Yeah, normally after this 56 days post-radiation, so basically we translate this as when a kid go under therapy, right? And then 56 days, at least in the mice life, will be when they're fully mature, right? And we observe at least an increase of 60% of fibrosis in those mice, yes. So it was a little bit, quite a lot.
And what was the percentage of change after exercise?
So super interesting. We didn't observe changes in the extracellular matrix accumulation after exercise, right? We didn't observe a decrease, but different studies have suggested that after exercise training, the collagen alignment is completely different, right? And also after exercise training, we also have an increase in a little bit in extracellular matrix accumulation that is held to the normal adaptation following exercise. What's really interesting not observe like actually a decrease in the ECM accumulation. However, one of the things that another master student is doing is to see that after exercise training, we observe a perfect alignment of collagen fiber and not like a match or whatever that could contribute to ECM accumulation and in pathological conditions. But very nice question there. We have to look at.
While people are thinking of more questions, I have a couple. So you induced or exposed only one limb to radiation, right?
Yep.
So were all of these changes only measured in that limb? I'm wondering how much like transfer or effect there is to other.
Yeah, everything was in one limb, but also we get the contralateral limb, right, as internal control. And also we did mice that were not irradiated, right? There is a different study from specifically Nicolas Collao lab that made the comparison between mice that never were irradiated and also the mice that were irradiated in one limb and also having all the analysis in the contralateral limb. The contralateral limb have exactly the same response than the mice that weren't irradiated. So basically we have our internal control there.
Okay, so your measures in the contralateral and your post measures in the contralateral arms.
Yeah.
And then one more question. Have there been studies that look at the effects of how much load or mass is part of these resistance endurance training protocols? Does that affect the kind of effect you see on the FAPs and the inflammatory markers, or I would assume there is?
Yeah, it's a really interesting question because there is a different models of resistant exercise training at least in mice, right? This is more similar to what is happening at the physiological level, but there is a model that is called synergistic ablation that we basically you remove gastrocnemius and you leave the plantaris just in the mice. And basically in 2-4 weeks, you have a massive hypertrophic muscle of the plantaris, right? It's not physiological because I'm not gonna go to you and cut you specifically the gastrocnemius to induce these hypertrophic conditions. And we try to do this the most physiological level as possible. Right, we took this specific protocol from Charlotte Peterson's lab that she have developed this since 2020, and we follow exactly the same protocol. But it's really interesting to know also to observe what is happening between the different loading regions, right? If this can affect specifically the FAP, but we haven't done it yet.
Venus.
You know, we are biomechanics here, so any mechanical testing, because the structure, the change in the structure, for example, of the extracellular matrix, the active force and all of that could have been shown in mechanical tests. So
Yeah. We didn't do any mechanical testing, but it was really interesting, something to do. I think this is just because we get a low number of kind of sample in this case, so we try to minimize all other type of testing, but will be something that really interesting to see that if the mechanical testing can change it after the treatment, after that specific endurance exercise training, but we didn't do.
did you look at the performance, like how long they can last?
Yeah. Actually, it was very interesting. They increase the performance running, and when we did sex difference, we know that actually females run way better than males, right? So that's why something that could be super interesting that also another postdoc in my ex lab that is really interesting in sex differences after training and also after these specific cancer treatments, she's looking into.
Were there any indications as to why those females had better?
No, I think so. If you see the literature, females, I don't know why run way better than males, and at least in mice. I don't know, Mauro, in rats too. You have any sex difference there in your results that potentially could lead to different regimen trainings. For sure, when you do these more specifically to males or to females. I don't have an answer for that.
Was there any difference in the post-radiation effects in between the sexes?
No.
Between males and females?
Pardon me. I didn't understand you with in vitro, like the cell culture.
Yeah.
Experiment, but did you have like satellite cells that are healthy and then you put them
Yes.
with maps that are, or
So basically,
paths that are either radiated or non-radiated, and then you saw the difference?
yeah, so basically, through facts of fluorescent activators are sorting, we isolate first our healthy muscle side cell from the contralateral limb. And also we irradiate from that contralateral limb, also we get our FAPs. And we took our FAPs from a radiated limb. So we have two healthy fabs and one healthy, for sure, muscle side cell. And then we let them grow, and we did this condition mediated experiment that we took the condition mediated experiment from the irradiated FAP, put it into the normal contralateral muscle side cell, right? And also from the contralateral FAP cells in turn control into this contralateral muscle side cell, and we see that difference.
And did you do the opposite as well, like satellite cells that are not irradiated, and then you put them with healthy fats after exercise and stuff like that to see like, is it the fats or the satellite cells themselves that are not able to differentiate and
Yeah.
produce good cells?
We haven't done yet. So it's really interesting to see what is happening vice versa, or for sure what is the crosstalk, not only through fabs to muscle side cell, but because we know that the release, for example, with one IL-6 or different cytokines to activate and differentiate this muscle side cell under hemostatic conditions, right? But we don't know what is the other crosstalk.
Then I think Mauro had a question.
Yeah.
Okay, we'll get to Mauro first and then I'll come back to you.
Okay. Regarding the muscle, maybe I missed it.
Yeah.
Did you test just the gastro?
Gastrocnemius. It's just a gastrocnemius because was the only limb that were injected with a randomized sarcoma, right?
But do you know if there is some muscle effect, the type of the fiber of the muscle could react differently to the radiation?
Yes, super interesting because I didn't show that. We thought in the beginning, and also we did all the mitochondria changes also based of, we thought in the beginning that follow radiation exposure, we're gonna have a decrease specifically in type 2 muscle fibers, right? However, we noted interestingly that it was type 1 muscle fibers, right? Were significantly lower after radiation exposure at long term. So that's why then we go and say, okay, maybe something is happening with the mitochondria. And we did another collaboration with another lab. What is happening with the mitochondria? We for sure observe that the mitochondria mechanistically, in that case, in the FAPs, go through more glycolytic metabolism instant to oxidative metabolism, right? And also we observe a mitochondrial fission in comparison to mitochondrial fusion. So basically, after radiation exposure, we observe this fragmented mitochondria that could lead to the loss of type 1 muscle fiber. Instant to type 2.
Go ahead, Venus.
Yeah. So in various muscle diseases, so have these cells been like studied, like for example, cerebral palsy, because we know that there is an increase in fibrosis. So do you know how they behave?
In cerebral palsy, haven't seen any paper. I haven't seen any papers yet. For sure, the Duchenne muscle dystrophy and other cardiac pathologies, they have been described, right? But never in cerebral palsy, I never seen a paper with this. For sure, it will be really interesting to know if they're the drivers of fibrosis in this context.
Is this research being followed up in humans, or are any of these results of your studies being applied to human cancer survivors, or?
Yeah, so, well, I show it to you, one of the small part of the result of the human ones. We're still trying to recruit more, for sure, to increase the N. Because in that case, we took muscle biopsy from the irradiation area and also the muscle biopsy from the non-irradiated area to try to compare. right, what is happening. In that case, it's a little bit difficult to all the consent when you are working with children, right? This is really difficult, but for sure would be really interesting to follow up all these changes and which is specifically the training modality better to mitigate this. Normally, when you have in those exercise guidelines, what is happening when you combine these two modalities or you have it separately. And mainly all the studies, as I mentioned to you before, are conducted in adult cancer survivors, leaving aside what is happening into the juvenile cancer survivors. So for sure, it will be interesting to see how which program of exercise training we can apply to the specific juvenile survivors can mitigate those long-term effects.
Well, if there are no other questions, then let's thank Dr. Nicolas Collao for his presentation.